Detailed explanation. Blood grouping is a crucial process in medicine, used to identify a person's blood type for safe blood transfusions, organ transplants, and understanding various health conditions. Let's dive into the details. What is blood grouping? Blood grouping involves testing a person's blood to determine which specific antigens are present on the surface of their red blood cells, RBCs. These antigens are markers that can trigger an immune response if the body recognizes them as foreign. Understanding antigens and antibodies. Antigens. These are proteins or molecules present on the surface of RBCs. In the ABO blood system, the main antigens are A and B. Antibodies. These are proteins found in the blood plasma. They are part of the immune system and attack foreign substances, including incompatible blood cells. The ABO blood group system. This system categorizes blood into four main types based on the presence or absence of A and B antigens on the RBCs. Type A has A antigens on the RBCs and anti-B antibodies in the plasma. Type B has B antigens on the RBCs and anti-A antibodies in the plasma. Type AB has both A and B antigens on the RBCs and no anti-A or anti-B antibodies in the plasma. Type O has no A or B antigens on the RBCs but has both anti-A and anti-B antibodies in the plasma. How to remember. Type A, antigen A, anti-B antibody. Type B, antigen B, anti-A antibody. Type AB, antigens A and B, no antibodies. Type O, no antigens, anti-A and anti-B antibodies. The Rh factor. The Rh factor is another antigen that can be either present or absent on the RBCs. The presence or absence of this antigen determines whether the blood type is positive or negative. Rh positive, Rh plus, RBCs have the Rhd antigen. Rh negative, Rh, RBCs do not have the Rhd antigen. Importance of Rh factor. Crucial in pregnancy. An Rh-negative mother carrying an Rh-positive baby can develop antibodies against the baby's blood. Important for transfusions. Rh-negative individuals should ideally receive Rh-negative blood to prevent immune reactions. Antisera and blood grouping tests. Antisera are solutions containing specific antibodies used to detect the presence of antigens on RBCs. Here's how it works. Anti-A serum contains antibodies against an antigen. If clumping, agglutination, occurs when mixed with blood, it indicates the presence of antigens. Anti-B serum contains antibodies against B antigen. Clumping indicates the presence of B antigens. Anti-D serum contains antibodies against the Rhd antigen. Clumping indicates the presence of the Rh factor. Steps in blood grouping test. Mixing blood with antisera. Blood samples are mixed with anti-A, anti-B, and anti-D antisera. Observing clumping. Anti-A clumping indicates A antigens. Anti-B clumping indicates B antigens. Anti-D clumping indicates Rhd antigens. Blood transfusion compatibility. Proper matching of blood types is essential to prevent immune reactions. Universal donor. Type O, no A, B, or Rh antigens. Universal recipient. Type AB+, no antibodies against A, B, or Rh antigens. Compatibility chart. O+, can donate to O+, A+, B+, AB+. O, can donate to all blood types. A+, can donate to A+, AB+. A, can donate to A+, AB, AB+. B+, can donate to B+, AB+. B, can donate to B, B+, AB, AB+. AB+, can receive from all blood types. AB, can donate to AB, AB+. Importance of donor history. Taking a thorough history of the blood donor is crucial for safe transfusions. This includes Medical history. Checking for any diseases or conditions that might be transmitted through blood. Previous transfusions. Ensuring the donor has not had adverse reactions before. Lifestyle factors. Assessing risk factors for blood-borne infections. Conclusion. Blood grouping is a fundamental aspect of medical practice, ensuring that blood transfusions are safe and effective. By understanding the ABO system, the Rh factor, and the role of antisera, we can prevent serious immune reactions and save lives. This knowledge is essential for anyone involved in medical care, from doctors and nurses to laboratory technicians and blood bank staff.